When I was diagnosed with HER2-positive breast cancer, I had one thought I couldn't shake. What if my diagnosis was wrong? What if they identified the wrong subtype? Or even worse, what if I don't even have cancer and I'm about to go for treatment that I don't even need? And I know I'm not the only person who has had that thought after receiving a cancer diagnosis. But breast cancer diagnoses are not just made from one quick look under the microscope. There are several biological tests that confirm what type of tumor it is. First, the pathologists examine the tumor tissue under the microscope. A standard stain called H&E highlights the structure of tissue and helps the pathologists to recognize the structure of cancer cells. And then doctors use special laboratory color dyes, and these dyes attach to specific proteins inside the tumor cells. And if those proteins are present, the cells change color under the microscope. For example, in HER2-positive breast cancer, the dye attaches to HER2 protein on the surface of a cancer cell. And when the tumor produces a lot of HER2, the staining becomes very strong. And also, pathologists actually look for something very specific. The stain highlights the cell membrane all around the cell. Almost like each cancer cell has its own brown outline. That pattern is one of the key signs of HER2-positive breast cancer. And if the staining results are unclear, the doctors have another special trick. They can look directly at the genes inside the tumor cell to see whether the HER2 gene is amplified. So the subtype of breast cancer is not just guessed from how the cells look. So a cancer diagnosis is not just someone's opinion under a microscope. It's confirmed through multiple layers of testing. And understanding that helped me feel much more confident about my breast cancer diagnosis.